Maintenance of culture. So now we have already discussed about the getting the discrete colonies of the required isolated colonies. So isolated organisms. Once we do that, we need to preserve it. Why should we preserve it? Because we require them in the viable condition. So once we put it into different media, it has to replicate again. It has to grow again. That is one reason. Second, we don't want any other organisms or any other contaminants to be present there, because when we are doing experiment, we require only one specific organisms and we can't differentiate by just seeing it. So we will always believe it is E. coli or it is Staphylococcus aureus. So if there is contamination, so it is difficult for us to work with it. So we don't require any contamination. So we need to avoid contamination. Third one is to stop the genetic mutations. So since these organisms are very single-celled and very primitive organisms, this undergo mutations very often to get adjusted to the environment, for the nutrient requirement, pH stabilities, temperature stabilities, etc. So this has to be stopped, because we have certain, like we say Staphylococcus aureus, it has its own genetic genome. We don't want it to change, so it will give different characters which will affect our experiment. So that should not happen. So we need to stop the genetic mutation, avoid the contamination, and which has to be in the living conditions or viable conditions. So that is the reason why we need to maintain or preserve the cultures. So how do we preserve the cultures? One easy or most often used method is refrigeration. So this you might have already used. So we have done many experiments where you have already refrigerated. So refrigeration is one of the oldest technique where we can preserve the agar plates, slants, broth cultures in the refrigerator at 4 degrees Celsius. So this will stop or bring the bacteria into static conditions, so that it will not replicate. Rather, it will just be living, or it will be in the dormant stage. So that will help us to preserve the bacteria. But this cannot be done for longer time. You know why? Because in the refrigerated conditions, the water will be taken up. That is how it is refrigerating. In any case, for that matter, any food or anything that happens to the media in the media present in the agar or the slants or the broth cultures. So the media will dry up, and the organism present in there, it is still living. So it will be taking up the medial nutrients in the media. So nutrients will deprive. So this will again, after a certain time, the organism will start dying because of the nutrient deprivation and no water conditions. So this is one of the problem in the refrigerations. So how can it be overcome? Is by periodic transfer to fresh media. So this is generally done in many laboratories. So what we do is prepare the media very often, and we will be transferring it periodically. Once we feel that media is getting dried up, we transfer the organism into freshly prepared sterilized media again, grow it at 37 degrees Celsius for overnight, or if it is fungi for 3 days, then we will again refrigerate it for 2-3 weeks. That is how we can maintain the cultures in the normal small laboratories. Then there is something called as paraffin method. So we know paraffin, mineral oils are all fatty contents. So this will help in preservation. So how it will help us? We generally use this for the slant cultures. We add paraffin to the slants. So this will create a oily layer. So the dehydration will be completely stopped. So the organisms will be living, but it can't replicate because the nutrient conditions are in the very less manner, and also the air it is in more of anaerobic conditions. So it is not allowing the organisms to replicate, rather it is just in the dormant stage. So it can be preserved in room temperatures only. Then there is something called freezing. So this freezing is a common technique again. So colder the temperature, the longer the viability of the cells. So this is the principle of the freezing. So we have already studied the temperature as a factor for bacterial growth. We know bacteria requires its own temperature for its growth. So why are we discussing about this freezing? Bacteria can't survive in the colder sections. We have already studied that. Why? Because there is ice formation, ice crystals formation. Once there is ice crystals formation, salt concentration will increase. Because there is no water, the water is converted into ice, the salt concentration will increase. This will disturb the activities of the cells. So cell will start dying. So we need to stop this. We need to control this. That will help us in the keeping the cells in the dormant stage. So we use something called as glycerol stock. So what is this glycerol stock as? We will add glycerol, 15 to 20% of glycerol to the organisms, and we will preserve it at minus 80, minus 90, minus 100 degrees Celsius. Generally it is minus 80 degrees Celsius. So what happens, why we are adding glycerol is, it is also called as cryopreservant or cryoprotectant, where it will protect the cells from the colder temperatures. So this glycerol is produced by many fishes, many organisms to protect themselves from the colder regions or colder temperatures. So this will help the organisms to survive in the colder regions, so by controlling the ice formation. So the glycerol will stop or control the ice formations. So thereby the salt concentration will be normal, and the organisms will be in the viable conditions, not very active, not very replicating conditions. So this can be used. Then there is something called as cryopreservation. You might have heard about liquid nitrogen, etc. So cryopreservation is one of the techniques used to preserve the organisms for longer time, like 15 years, 20 years, etc. So same glycerol will be added as a cryopreservant and will be stored in liquid nitrogen. So liquid nitrogen will be at minus 130 to minus 190 degrees Celsius. So in between we can adjust the temperature and preserve the cells in that condition, so for a longer time. But we have to take care that liquid nitrogen should not enter inside the cells, inside the culture, because the liquid nitrogen is again toxic to cells. So the liquid nitrogen should be just present outside in the environment, so it will form a colder temperature. To avoid the entering of liquid nitrogen, there is something called as gas fumes of the nitrogen, which is used in place of the liquid nitrogen. But the refrigerators which use this gas nitrogen is really costly. So that is one of the negatives about the liquid nitrogen preservation. These glycerol stocks or liquid nitrogen preservations will be done in higher research laboratories, where they will be storing the organisms for really longer time. Then there is something called as lyophilization. So what is lyophilization is just freeze drying. So what we will be doing is, here we will be adding lyoprotectant like sucrose solutions to the organism solution, and since there is sucrose, the organisms will be protected from the freezing. Or once we start freezing, all the water content present in the solution will be completely taken out by vacuum. So once all the water is taken out, it will be like a powdery thing. So once this powdery thing it happens, it can be stored for a longer time, and this will help us for the transportation of the organisms. So most of the transportation of organisms will take by lyophilization. Here there is we use something called as sublimation process. You know what is sublimation? Conversion of water directly into the solid directly into the gas conditions. That is, ice will be directly converted into gas. That is the reason why it is called as freeze drying. So this will be used in the production laboratories, drug productions, etc., to preserve the drugs and the vaccines, etc. And it will be used for transportation of the organisms, has to be oftenly done. So these are few techniques where we can preserve the organisms for really long times. This refrigeration and periodic transfer of fresh media will be in the smaller laboratories for less than 1 year or less than 1 month time. The freezing and the cryopreservations will be for 15 years long. This is for transportation and so on. There are different techniques used for different purposes, so we will use the technique based on the what requirement the experiment is doing. So that is one thing we will be doing. So I have attached a link for the lyophilization. You can just go through that, which you can understand how the lyophilization is done.